welcome to my world, Biopulse. Let me ask you something interesting. If I ask you, what is the smallest unit of life? What would be your answer? Yes, that's right. The answer is cell. Your body has trillions of cells, but did you know that every one of these cells is like a tiny factory that producing energy, making protein, storing informations, and even communicating with other cells also. In fact, your entire body started from just one single cell, that is zygote. So, how does such a microscopic structure control all the activities of life? What are the tiny machines inside that keep us alive every second? Today, we are going to enter an amazing microscopic world of life and will understand why it is called the unit of life. So, let's begin. Comes the first question. It's about cell structure and function. First question, in living organisms, detailed description that brings out their knowledge of diversity is about option A, their form, option B, their appearance, option C, both, option D, none. And then here the right option is option C, both. Both in the sense their forms and then their appearance. Next question. Question number 2. What brought out the unit of diversity, the cellular organism of all life forms? Option A, theory of evolution, B, species theory, C, cell theory, D, Darwinian theory. And then here right option is C, cell theory. Cell theory is the one of the unit it brought actually what actually the unit of diversity means, how actually the diversification is going in the world of the living organism. That's what cell theory is, the main theory we can consider. Question number 3. What is not true about physico-chemical approach? Not true, he is asking about the physico-chemical approach. Option A, established by analysis of living tissue for element and then compounds. Option B, explains what type of organic compound is present in living organism. C, explains the abnormal process that occurred during any diseased condition. Option D, this approach is known as forward biology. So here the right option is option D. This approach is known as forward biology. Fourth question. Here comes actually the question starts. Fourth question. Unicellular organisms are capable of option A, independent existence, B, performing the essential function of life, C, both, D, doesn't ensure independent living. Unicellular organisms are capable of these two. So the right option is option C, both. Fifth question. Living cell was firstly seen and described by living cell. Okay, living cell was firstly seen and described by Robert Hooke, Anton von Leeuwenhoek, Robert Koch, Robert Brown. And the right answer is option B, Anton von Leeuwenhoek. Questions from cell theory. Sixth question. Cell theory was proposed by A, Matthias Skleden and Theodor Schwann, B, Skleden and Schwann and Virchow, C, Rudolf Virchow, D, Sutton and Boyer. The cell theory was proposed by Matthias Skleden and then Theodor Schwann. Don't get confused here. This is the final answer. Okay, Rudolf Virchow is modified the cell theory. Seventh one. All the plants are composed of different kinds of cells which forms the tissue of plant. This statement was given by all the plants are composed of different kinds of cells. Yes, of course. Which forms the tissue of a plant. This statement was given by a German botanist Rudolf Virchow. B. A British zoologist Matthias Clidden. C. A British zoologist Theodore Swan. D. A German botanist Matthias Clidden. And then here the right option is option D. A German botanist Matthias Clidden. Eighth question. Who studied the different types of animal cells to propose cell theory? Option A. A British zoologist Matthias Clidden. B. A German botanist Theodor Swan. C. A physicist Rudolf Virchow. D. A British zoologist Theodor Swan. And then here the right option is option D. A British zoologist Theodor Swan. He studied about the animal cells. And then whereas Matthias Clidden was a botanist who studied about the plant. Ninth one. A thin outer layer studied by Theodor Swan, nowadays known as a thin outer layer. Nowadays known as option A. Plasma membrane. B. Cell wall. C. Glycocalyx. D. Middle lamellae. And then here the right option is option A. Plasma membrane. Question number 10. Based on studies of Matthias Clidden, what is a unique character of plant cell? Based on Clidden's experiments, what is a unique characteristic of plant cell? Option A. Cell wall. B. Middle lamella. C. Glycocalyx. D. None of this. According to him, after his experiment, he said that plant cell has a unique characteristic, that is the presence of cell wall, which is only the most unique characteristic for what? Plant. So that is the answer. Question number 11. The hypothesis that the bodies of animals and plants are composed of cells and their products was proposed by, means what? Animals and then plants are composed of cells, of course, and their products. Cells and cells products in the sense cell, whatever cell is going to produce like proteins, lipids, etc, etc, etc. So this was proposed by A. Clidden and Swan. B. Rudolf Virchow. C. Swan only. D. Virchow and Clidden. So here the right option is A. Clidden and Swan. They proposed the cell theory. There they mentioned the cell or else the bodies of the animal and plant are composed of cell and their products. 12th question. Scientist who gave the final shape to cell theory. Who gave the final shape to cell theory? Option A. Clidden. B. Swan. C. Virchow. D. Clidden and Swan. Keep in mind, the cell theory was proposed by Clidden and Swan, that's clear, but it was finally shaped by giving omnis cellulae e cellulae statement was definitely by Virchow. So here the right answer is Virchow. Okay, Rudolf Virchow, who is he helped a lot to finalize the shape of the cell theory. Question number 13. Which of the following is related to cell theory? All living organisms are composed of cells and products of cells. Second one, proposed by Clidden and Swan. Third one, modified by Rudolf Virchow. Fourth one, all cells arise from pre-existing cells. Fifth one, omnis cellulae e cellula. So here among these all, every sentence is related to cell theory only. So the right option is all 5. An overview of cell. 14th question. What is the delimiting boundary around a human chick cell? Boundary of the human chick cell. Option A. Cell membrane. B. Protoplasm. C. Protoplast. D. Cell wall. And then here the right option is cell membrane. The boundary of the cell. 15th one. What is the semi-fluid matrix inside the cell? Semi-fluid matrix inside the cell. Option A. Cell membrane. B. Protoplast. C. Cytoplasm. D. Nucleus. Here the right option is cytoplasm. Cytoplasm is the semi-fluid matrix which is filled inside the cell, whereas all the cell organelles were present or else embedded in the that fluid content, semi-fluid content. 16th question, how many of the following statements are not true? Statement 1, all cells have membrane bound nuclei and nucleolus. All cells have membrane bound nuclei and nucleolus. 2, nucleus contains chromosome. 3, DNA is genetic material. 4th, cytoplasm is the main area or else arena of cellular activities in plant and then animals. And then here the right option is option A, only 2, 3 and then 4 are. Make changes here. Here how many of the following statements are not true means only first one. Only one is not true, that is option D is right. Okay, all cells have membrane bound nuclei and nucleolus, but yes, of course, but except RBCs and in situ tube of the plant cell, so there is no nucleus, they are the lack of nucleus, so this statement is not true. Only only one is the correct one for this, so please make a correction there, sorry for that. 17th one, besides the nucleus, the dash cell have other membrane bound distinct structure. Beside the nucleus, the dash cell have other membrane bound distinct structures. Option A, eukaryotic. Option B, prokaryotic. Option C, both A and B. D, none of this. Beside the nucleus, when it is called about the beside the nucleus, you must go with the eukaryotes, because in the prokaryotes nucleus won't be there. 18th one, what is the non-membranous organelle present in both eukaryotic and as well as prokaryotic cell? A, endoplasmic reticulum. B, protein. C, mitochondria. D, ribosome of ATS type. See here, what is non-membranous organelle present in both eukaryotic and prokaryotic, he is asking. This is endoplasmic reticulum is membranous, protein is not at all organelle, and mitochondria is membranous, so the right option is ribosome of ATS type. 19th one, animal cell have another non-membrane bound cellular organelle known as, animal cell has another non-membrane bound organelle known as option A, microbodies. B, nucleus. C, lysosome. D, centrosome. Here the option is D, centrosome, because all of the other are made up of membranous bound cellular organelles. So the right option is centrosome. 20th question, which of the following is not incorrect? Which of the following is not incorrect? A, mycoplasma is the smallest cell, 0.3 micrometer in width. B, bacteria could be 3 nanometer to 5 nanometer in length. C, human RBCs are about 7 mm in diameter. D, cells shape is independent of their work they perform. So here, which one is not true? That is not incorrect. Means, which of the following is not incorrect? Among this is mycoplasma is the smallest cell, that is 0.3 micrometer in width. Prokaryotic cell. 21st question. The prokaryotic cells are represented by option A, bacteria. B, blue-green alga. C, mycoplasma, and then PPLO, pleuropneumonia like organism, and then D is all of these. So here comes the right answer, that is the D, all of these. 22nd question. All prokaryotic cells have this cellular boundary surrounding the cell membrane except in mycoplasma. That is option A, glycocalyx, option B, protoplast, option C, cell wall, option D, cytoplasm. So here the right option is answer C, that is cell wall. Cell wall is present cellular boundary surrounding the cell membrane. Mycoplasma won't have the cell wall. 23 question. Which of the following is related to prokaryotic cell? Hey, option A, have no well defined nucleus. B, have basically naked genomic material. C, in addition to genomic DNA, the extra genomic DNA is also present, known as plasmid. And then D, all of the above. And then here the right option is option D, all of the above. These all are related to prokaryotic cell. 24. Which of the following confirms certain unique phenotypic characteristics to the some bacteria? Option A, chromosomal material. B, extra chromosomal material. C, mitochondrial DNA. D, genetic material present in chloroplast. So here the right option is B, extra chromosomal material, which is going to be called as a plasmid. Plasmid is a unique phenotypic characteristic of some bacteria which is going to be used in the biotechnology. 25th question. A special form of cell membrane which is the characteristic of prokaryotes is option A, plasmid, B, cell wall, C, cell membrane, and then D, mesosomes. So here the right option is the mesosome. Mesosome is the infolding of cell membrane inside the cell, which is helpful for the respiration of the prokaryotes. So next question, that is 26th one. Which of the following is membraneless bodies other than ribosomes? Membraneless bodies other than ribosomes. Option A, cell wall. B, inclusion. C, mesosomes. D, chromatophores. And then here the right option is B, inclusion bodies. 27th one. Which of the following is the essential infoldings of cell membrane? Option A, inclusion. Option B, mesosome. Option C, chromatophore. Option D, plasmid. So here the right option is option B, mesosome. Next question. See, let me show you where actually the infoldings is. See, this is the cell membrane, and the cell membrane is showing you the inner folding. This inner folding itself is called what? Mesosome. Okay now. Next question, that is from cell envelope and its modification. 28. What is the sequence of cell envelope in most of the prokaryotic cell, outer to inner one? Option A, glycocalyx, cell membrane and cell wall. Option B, cell membrane, cell wall and glycocalyx. Option C, cell wall, glycocalyx and cell membrane. Option D, glycocalyx, cell wall and cell membrane. And then right option is glycocalyx, cell wall and cell membrane. Next one, 29th one. The prokaryotic cell have a single protective unit made up of option A, glycocalyx plus cellulosic cell wall plus cell membrane. B, peptidoglycan cell wall plus cell membrane plus glycocalyx. C, chitinous cell wall, cell membrane and glycocalyx. D, siliceous cell wall, glycocalyx plus cell membrane. Here the right option is peptidoglycan cell wall plus cell membrane plus glycocalyx. 30th question. How many of the following statements are correct? Statement 1. Glycocalyx is outermost layer. 2. All three layers have same function. 3. Bacteria can be classified on the basis of difference in the cell envelope. Fourth one, bacteria can be classified on the basis of response to the staining procedure. So here, how many are the correct one he is asking? So here there are only 3. Glycocalyx is outermost layer is correct. All 3 layers have same function is wrong. Third one, this one is correct, and then fourth one is correct. So only 3 are correct among them. 31. The bacteria that take up gram stain are gram positive type, B gram negative type, C both type, D neither gram positive nor gram negative. Here the right option is gram positive type. 32. The bacteria that do not take up gram stain are A gram positive, B gram negative. Just now we saw if it is taking the gram stain, then it is said to be a gram positive. Then if it is not taking, obviously it will be gram negative type. Be smart work, don't do a donkey work. Okay. 33. Which of the following in a bacteria envelope is a loose sheath of slimy layer? Option A glycocalyx, B cell wall, C cell membrane, D none of the above. Slimy layer, which is the loose sheath, is made up of what? Glycocalyx. It is called as what? Glycocalyx. So here the right option is option A. 34th question. Glycocalyx could be a thick or tough layer and known as, if it is thick and then tough, then it is known as option A slime layer, no. Option B cyst, no. C capsule, yeah, it's the right option. If it is loosely arranged, then it is called as slimy layer. If it is arranged very thick and then tough, then it is called as what? Capsule. 35th question. Which of the following determines the shape of the bacterial cell? Which of the following is determine the shape of the bacteria? Means option A glycocalyx, B capsule, C cell membrane, and then D cell wall. And then here the right option is option D cell wall. 36th question. How many of the following following is not incorrect regarding a cell membrane in prokaryotes? First statement, selectively permeable in nature. Second, structurally similar to eukaryotic cell. Third one, interact with outer world. Fourth, innermost layer of cell envelope. Fifth, living layer. And then here the right option is only 1, 4 and 5th one are not incorrect. 37th question. How many of the following is the membranous extension into the cell of bacteria? The membranous extension of the bacteria. First one, mesosome, yes. Tubules, yes. Vesicles, yes. Lamellae, yes. Chromatophore, no. Inclusions, yes. So here the right option is option C, 5. Rather than the chromatophore, other 5 are the infoldings. Not only infoldings, they are the membranous extensions of the bacteria, I can say. Next question, that is question number 38. How many functions from the following the mesosome can perform? Number 1, DNA replication, of course. 2, respiration, yes. 3, DNA distribution to the daughter cells, of course. 4th one, secretion. 5th one, increases surface area, yes. 6th one, contains enzymatic content. So all the 6 are correct here about the mesosome function. So the option C is correct here, all of the 6. 39th one. In cyanobacteria, there are some another membranous extensions except mesosome are A. Inclusions, B. Fat globules, C. Chromatophores, D. All of the above. Here the option is C. Chromatophores. 40th one. What are structures related to bacterial flagellum? What are the structures related to bacterial flagellum? Option A, basal body and filament. B, basal body, hook and filament. C, hook and filament. D, filament only. So here the right option is bacterial flagellum has basal body, hook and then filament. So the right option is option B. See, this is the structure of the flagellum. This is the basal body. This is the hook and this one is the filament. Next, 41th. Longest portion of flagellum is option A, basal body, B, hook, C, filament, D, none of the above. You can say which one is the longest one. This is the filament. Okay, here somewhat mistake has happened. This is the filament, this is the hook and this one is the basal. By seeing the diagram only you can answer for this question. The longest portion is filament. 42. Which of the following structure helps in motility in the bacterial cell? Option A, cell membrane, B, pili, C, fimbriae, D, flagella. So here the right option is D, flagella. 43. Which of the following is not a surface structure? A, fimbriae, B, pili, C, flagella, D, inclusion. So here D, inclusion is not a surface structure, it is they are present inside the body of the bacterial cells. 44. Which of the following is small bristle like fibers sprouting out of the cell? Which one is bristle like and then they are very small? Option A, pili, B, cilia, C, flagella, D, fimbriae. Here the right option is fimbriae. So next question, that is 45. Which of the following is elongated tubular proteinaceous structure? Option A, pili, B, inclusions, C, mesosomes, D, fimbriae. Inclusion body is not an elongated one and then they are granularly present in the inside the body of the bacteria. C is infolding of cell membrane which is inside, and then fimbriae is smallest one, just now we discussed. So that the right option is pili, which is the elongated one, tubular which is proteinaceous. 46th question. Which of the following helps the bacteria attach to the rocks in streams? A, inclusions, B, mesosomes, C, fimbriae, D, pili. Here the right option is fimbriae. Questions from ribosomes and inclusion bodies. 47th question. Ribosomes are associated with the structure in a bacterial cell. Option A, tRNA strand, B, Golgi body, C, cell membrane, D, ER, that is endoplasmic reticulum. Here the right option is cell membrane. Ribosomes will attach or else associated with the cell membrane in the bacterial cell. 48th one. Ribosomes in bacterial cell are option A, 20 nanometer to 30 nanometer in size, B, made up of two subunits, larger one is 60S and then smaller one is 40S, C, made up of two subunits, larger one is 50 and then smaller one is 30, D, associated with ER and cell membrane. Here the right option is option C. They made up of two subunits, larger one is 50 and then smaller one is 30S. Next question, 49th one. A polysome is polysome. Poly in the sense many, here rome refers to the ribosome. So on the basis of the name itself, you can just guess. Question number 49, first option. Several mRNA bound to a single ribosome. B, several subunits of ribosome attached to each other. C, several ribosomes attached to a single strand of mRNA. D, several mRNA attached to each other. So here the right option is option C, several ribosomes attached to a single strand of mRNA. So this is the polyribosomes. See, this is 1, 2, 3, 4, 5, 6, 6 ribosomes which are attaching to a single mRNA. At a time, at the same time they produce what? Proteins. Different kind of proteins are going to produce by the polyribosomes. 50th question. Which of the following structure translate the mRNA into proteins in a bacteria? Option A, inclusions of cytoplasm. B, ribosomes of ER. C, ribosomes of polysome. D, polysomes of ribosomes. So here the ribosomes of polysome is correct. The right option is ribosomes of polysome. 51. Inclusion bodies in a prokaryotic cell are A, reserve material containing structure. B, cell membrane enfoldings. C, membrane bound structure. D, all of the above. Here the right option is they are the reserve material containing structure. Which one? The inclusion bodies. 52. Which are examples of inclusion bodies? First one, mesosomes. Second one, chromatophores. Third one, gas vacuoles. Fourth one, phosphate granules. Fifth one, synophysin granules. Sixth one, glycogen granules. Inclusion bodies are in the form of granules. Mesosome is the enfolding. Chromatophore is not the what we want. Gas vacuole is one of the type of the vacuole. So here the right option is what? Only 3, 4th, 5th and 6th one. These are what? They are the inclusion bodies. Okay. Next, 53. Inclusion bodies can be found in all types of cells. B, all eukaryotic cells. C, blue-green algae and green photosynthetic bacteria. D, prokaryotic only. So here the right option is prokaryotic only. Inclusion bodies are present in prokaryotic cells. Now the questions from eukaryotic cells. 54. All of these are eukaryotic except A, protista, B, plantae, C, monera, D, animals. So here the right option is monera. 55. How many of the following statements are true regarding eukaryotic cell? Statement 1. Cytoplasm has extensive compartmentalization. 2. Presence of membrane bound organelles. 3. Organized nucleus. 4th, a variety of complex locomotor and cytoskeletal structure. 5th, genetic material is organized into chromosomes. So here the right option is option 5, that is option D, 5. All the 5 are correct. Next, 56. Statement 1. Plant cell differs from animal cell. Statement 2. The former one possesses cell walls, plastids and a large vacuole which is absent in later one. So here the right option is option A. Both the statements are correct. 57. Centrioles are present in A, animal cell, B, plant cells, C, both animal and plant cell, D, all other than plant cell. Here the animal cell. Centrioles or the centrosomes is a unique character which is only related with the animal cell. Animal cell has the centrosome whereas plant cell doesn't have. 58th question. Which of the following correctly explain the diagram? Here the diagram is given and then these are the parts which we must identify them. Okay, here the first one is chloroplast and then second one which is indicating the nucleus and then third one is Golgi and fourth one is rough endoplasmic reticulum. So here first one chloroplast, first answer itself is A. Now the questions from cell membrane. 59th one, the detailed structure of membrane was studied. The detailed structure of the membrane was studied. Option A, only after the advent of the electron microscope in 1950s. B, unable to deduce the possible structure of plasma membrane. C, both, and then D, none. Here the option A and then B both are correct with respect to the cell membrane structure study. 60th question, which of the following cell study enables the scientists to deduce the possible structure of plasma membrane? Option A means among them which is helpful for to study the plasma membrane structure in the sense RBC, that is red blood cells, cork cells which is the dead one, and then WBC is white blood cells, and then bacterial cell. Here the best and then foremost material for the study of the structure of plasma membrane is RBC. 61. Cell membrane is mainly composed of option A, lipids and proteins, B, proteins and cholesterol, C, lipids and carbohydrates, D, carbohydrates and proteins. And then here the right option is lipids and proteins. Our cell membrane is mainly composed of lipids and then proteins. 62. Which is correct arrangement of lipid molecule in the cell membrane? Lipid molecule in cell membrane. Option A, polar head outside, non-polar tails inside. B, non-polar head outside, polar tail inside. C, polar tail outside, non-polar head inside. D, polar tail inside and non-polar head outside. So here the right option is option A, polar head is outside and the non-polar tail will be inner side, so that they can be protected. See here, this is the fluid mosaic model where you can see the lipid bilayer. This is the lipid bilayer where the polar head is outside and then tail. This is the non-polar tails which are inside. Okay, now this is the polar head and then this one is the tail, which polar head is outside. This is also polar head, this is also polar. Bilayer in the sense two layer will be there, no? So this two head, this is one lipid layer, a lipid bilayer, and this one is one lipid bilayer. Here this is this side is outside and this one is this side outside. So the tails are inside and then heads are outside. This is what about the lipid bilayers. Next one, 63. What ensures that the non-polar tail is protected from aqueous environment? Option A, polar head outside, non-polar tail inside. Option B, non-polar head outside, polar tail inside. C, polar tail outside, non-polar head inside. D, polar tail inner side, non-polar head outside. So just now I explained about that, which is because a polar head is outside, which is helpful to protect the inner side, that is non-polar tails, because the non-polar tails are hydrophobic. They are hydrophilic. Polar head are hydrophilic and the non-polar tails are hydrophobic. 64. The constituents of cell membrane are phospholipids, carbohydrates, proteins, cholesterol and phosphoproteins. Among them, which are constituent of cell membrane means phospholipids, correct, carbohydrates, correct, proteins, correct, cholesterol is also correct but not the phosphoproteins. 65. Which of the following study revealed that cell membrane also contains proteins and carbohydrates? Option A, electron microscopic study. Option B, phase contrast microscopic study. Option C, biochemical investigation study. D, cobalt chloride paper test study. Here the right option is biochemical investigation study. 66. Which of the following statement is incorrect? A, the tail is hydrophobic of saturated hydrocarbons. B, The tail is hydrophilic of saturated hydrocarbons. C. The tail is hydrophobic of unsaturated hydrocarbons. D. The tail is hydrophilic of unsaturated hydrocarbons. Here the right option is the tail is hydrophobic. Just now I said here tail in the sense the lipid bilayer tail. Okay, hydrophobic and it is saturated hydrocarbon. 67th one. Which of the following statement is incorrect? A. The ratio of proteins and lipids varies considerably in different cells. B. In erythrocytes, it has approximately 52% of proteins and then 40% of lipid. This statement is correct. And then option C. On the basis of ease of extraction, membrane protein are of extrinsic and intrinsic type. Yes, of course. None of the above. Here the right option is option A. The ratio of proteins and lipids varies considerably in different cells. This is actually incorrect. 68th one. The improved model of structure of cell membrane was proposed by improved model. A. Missels and Starr. B. Sklidden and Swan. C. Anton von Leeuwenhoek. D. Singer and Nicholson. The model itself is given Singer and Nicholson model. So here the right option is Singer and Nicholson. 69. The squashy fluid nature of lipid enables A. Flip-flop movement of proteins within the lipid bilayer. B. Lateral movement of proteins within the lipid bilayer. C. Flip-flop movement of lipid crossing the protein bilayer. D. Lateral movement of lipid crossing the protein bilayer. Here option B. Lateral movement of protein within the lipid bilayer, which will be helpful for the squashy fluid nature of the lipid. 70th one. One of the most important function of the plant membrane is A. Transport of molecule across it. B. Flip-flop movement. C. Secretion. D. Cell enlargement. The most important function of the plasma membrane is transport of the molecule, which is selectively permeable. 71 question. What ability explains the fluidity of cell membrane? A. Squashy fluid nature of cell membrane. B. Lateral movement of protein. C. Cell growth formation of intercellular junctions, secretion, endocytosis and cell division. So here the right option is all of the above. These all are about the ability to explain the fluidity of the cell membrane. 72. The plasma membrane is option A. Semi-permeable in nature. B. Impervious in nature. C. Impermeable in nature. D. Selectively permeable in nature. So here the right option is selectively permeable in nature. 73. How many of the following functions the cell membrane can perform? Active transport, osmosis, passive transport. So all the cell membrane can perform active transport also, osmosis also, passive transport also. So here the right option is option C, all. 74th. Sodium and then potassium pump, sodium potassium pump transport molecules. Option A by passive, B by active transport, C by utilization of ATP, D by both B and C. So here by active transport, potassium pump theory, sodium and potassium pump theory is there, no, where we can study the how actually the potassium and then sodium are going to be exchanged within the cells. Here onwards the questions are from cell wall. 75. The outer covering of fungi and then plant is A. Glycocalyx, B. Cell wall, C. Cell membrane and then D. All. So here the option B, cell wall, is correct. The outer covering of this fungi. 76. What is the function of cell wall? A. gives shape to the cell, B. protects the cell, C. cell to cell interaction, D. all of the above. So option D, all of the above is correct. 77. What are chemical composition of algal cell wall, that is cellulose, galactans, mannans, calcium carbonate and chitin? Here the right option is only 4 of them. Chitin is not present in the algae. 78. Cell wall of plants consists of option A. cellulose and pectins only, B. cellulose, hemicellulose and pectin only, C. cellulose, hemicellulose, pectin and proteins, D. hemicellulose and proteins. Here the option C, it contains cellulose, hemicellulose, pectins and proteins. 79. Which of the following is capable of growth? Which of the following is capable of growth? Option A. primary cell wall, B. secondary cell wall, C. tertiary cell wall, D. all of them. So here the right option is A. primary cell wall. 80th question. Secondary cell wall is formed outside the primary cell wall, inside the cell membrane, inside the plasmodesmata, inside the primary cell wall. So here the right option is D, inside the primary cell wall. Secondary cell wall will form inside the primary cell wall. 81. Which of the following in plant act as glue between the neighboring plant cells? Calcium pectate, magnesium pectate, calcium and magnesium pectate, none of the above. Here the right option is calcium pectate, which is present in the middle lamellae. This is the middle lamellae. This is one cell and this one is one more cell. Both the cells are going to be attached with the help of a glue like structure, which is said to be what? Middle lamellae, which is rich in pectin. 82. Which of the following is transversed by plasmodesmata? A. cell wall and cell membrane, B. cell membrane and glycocalyx, C. cell membrane, cell wall, glycocalyx and middle lamellae, D. cell wall and middle lamellae. Here the right option is cell wall and middle lamella. See, this is just a minute, I will show you. Yeah, see, this one is one cell and then this one is one cell. Here this is the opening, which is said to be what? Plasmodesmata, which is helpful for what? Movement of molecules across the cell. This one is one cell and then this one is one more cell. This is the small pores, small openings, which is said to be what? Plasmodesmata. Next, questions from endomembrane system. 83. What are the constituent of endomembrane system? Option A. endoplasmic reticulum, B. Golgi body and endoplasmic reticulum, C. Golgi body, lysosome and vacuole, D. Golgi body and lysosome. Here the right option is option C, Golgi body, lysosome and vacuole. Why they are said to be endomembrane system? Because they perform the function accordingly. Trick to remember this: Golgi, lysosome and vacuole. Every girl loves vacation. Every girl loves vacation. Here E means G means Golgi, and then love means lysosome, vacation means vacuole, represents this one. Okay, you just keep in mind this sentence, you will remember the endomembrane system. Golgi, lysosome and vacuole. Every girl loves vacation. 84th one. Why mitochondria, chloroplast and peroxisomes are not the part of endomembrane system? Because A. they are autonomous organelles, no. B. they are semi-autonomous organelles, no. Because they are not coordinated with the endomembrane system. This one is the right one. 85. Which of the following is the network of tiny tubular structure scattered in cytoplasm? A. B. Golgi body, C. lysosome, D. vacuole. So here the right option is A, ER, which is scattered in the cytoplasm. 86. Which of the following structure divides the intercellular space into two compartments? Intercellular space into two. Even though within a cytoplasm also it is making intercellular space. That one is ER. It gives intercellular space into two compartments. Okay, that is extraluminar and then luminar component. 87th question. The extraluminar and luminar compartment represents A, cytoplasm and inside the ER, B, inside the ER and cytoplasm, C, outside ER and cytoplasm, D, cytoplasm and outside the ER. Here the right option is cytoplasm and inside the ER. 88th one. The ER having ribosomes attached to its outer surface is known as ER having the ribosomes. Then it is said to be what? Rough endoplasmic reticulum. Because of the presence of the ribosome only, the ER is having two types. One is RER and one more is SER. If ribosome is present, it is said to be RER. If ribosome is absent, then it is said to be SER. See, this is the ribosomes which are attached to the endoplasmic reticulum. That's what it is said to be, a rough endoplasmic reticulum, which is helpful for the synthesis of protein. 89th one. RER is frequently observed in cells actively involved in A, protein synthesis, B, lipid synthesis, C, DNA synthesis, D, glucose synthesis. Right option is option A, protein synthesis. 90th one. Which of the following is continuous with the outer membrane of nucleus? A, RER, B, SER, C, Golgi body, D, lysosome. Here the right option is option A, RER. 91. Steroidal hormones are synthesized by A, RER, B, lysosome, C, SER, D, ribosome. Here the right option is SER. See, this is the smooth endoplasmic reticulum where the ribosome is absent. 19.2. Golgi body was firstly observed by option A, Camillo Golgi in 1898. This one is right. 93. Golgi body is reticular structure, densely stained structure made up of cisternae, tubule and vesicle, concentric cisternae. So here the right option is all of the above are correct regarding the Golgi. This is the Golgi apparatus where you can see the vesicles and then cisternae. This is the cisternae and then this is the vesicles. 94. What is the diameter of cisternae of Golgi body? What is the diameter of cisternae of the Golgi body? 0.5 micrometer to 1 micrometer, 0.1 micrometer to 2 micrometer, 0.2 micrometer to 2.5, 3 to 2 micrometer. Here the right option is 0.5 to 1. 95. The convex face of cisternae of Golgi body is also known as A, cis phase, 2, forming phase, 3, trans phase, 4, maturing phase. And here the right option is option A, cis phase. It is said to be what? Cis phase. And the next question, that is 95. The convex face of cisternae of Golgi body is also known as, this is the convex face, cis phase it is called. Okay na? 96th one. Which of the following statement is correct? Cis and trans phases are same but interconnected. B, cis trans phase different and not interconnected. C, cisternae is 0.1 to 2 micrometer in diameter, and then D, none of the above. Here the right option is option B, cis and trans phases different and not interconnected. Just now I showed you this one. Cis phase is here and then trans phase is here. They are not interconnected. So the option B is right. 97. Golgi body principally perform the function of A. Secretion, B. Packaging of materials, C. Both, and then D. None. Here the main function of the Golgi is packaging. 98. Materials to be packed in the dash fuses with the dash phase. Option A. Cis phase and trans phase. Option B. Trans phase and cis phase. C. ER and cis phase. D. ER and trans phase. Here the right option is option C. ER and trans phase. After the synthesis of protein from the ER, they are going to be taken by the cis phase and then transported out, packed, and then it is going to be transported. 99th question. A number of proteins synthesized by ribosomes on the dash are modified in the dash of the dash. Option A. ER, Golgi body and cisternae. Here the right option is option D. Here the ER are modified in the form of cisternae of the Golgi body. 101 century question. The vesicular structure formed by the process of packing in the Golgi apparatus is A. Vacuole, B. ER, C. Lysosome, D. All. Here the right option is lysosome. 101. The isolated lysosomes vesicles have been found to be very rich in lipases, proteases, carbohydrates. So here the right option is all. They all are isolated lysosome vesicles only. 102. Enzymes present in lysosomes are accumulatively known as acid proteases, lipases, acid hydrolases and carbohydrates. Here the right option is acid hydrolases. 103. The membrane bound space in cytoplasm is known as ER, Golgi body, lysosome and vacuole. The membrane bound space inside the cytoplasm is known as what? Vacuole. This is the vacuole. The vacuole's membrane is said to be what? Tonoplast. 104. Vacuole contains hydrolases, lipases, proteases, water, sap, excretory products and materials not useful for the cell. So here all of them are correct about the vacuole. 105. The membrane of vacuole is option A. Single membrane, B. Tonoplast, C. Both, and then D. None. Here the right option is both. It is made up of single membrane which is called as what? Tonoplast. 106. In a plant cell, vacuole can occupy up to dash percentage space of cell. A 70, B 80, C 90, D 50. Here the right option is C. 102.7. In plants, tonoplast facilitates the transport of number of dash the concentration gradient ions and against the concentration gradient. Option C. 108. How many of the following statements are not wrong? Concentration of same ions inside the vacuole is significantly higher. 2. In amoeba, contractile vacuole helps in osmoregulation and excretion. 3. In protistans, food vacuoles are formed by engulfing the food particles. So among them all three are correct. Number 109. Which of the following statement is correct about mitochondria? Option A. Easily visible under the microscope without stain. B. Number of mitochondria per cell is invariable. C. Number of mitochondria depends on the physiological activity of a cell. And then D. All of the above. So here the right option is option C. Number of mitochondria depends on the physiological activity of the cell. If the cell's activity is more, then it is in need of more mitochondria. So the number of mitochondria will be more in such kind of cells. For example, muscle cells. Next, 110. How many of the following statement is correct regarding mitochondria? Among them, how many are correct? 1. A sausage shaped structure. 2. Diameter is 0.221 micrometer. 3. Average diameter is 0.5 micrometer. 4th, length is 1 to 4.1 micrometer. So here the right option is 4. All the 4 are with respect to what? Mitochondria only. 111. Each mitochondria is dash membrane bound structure dividing its lumen into dash distinct compartment. Here what is X and then what is Y we should mention. Each mitochondria is X single and 1, X is double and then 1, X is single and then 2, D is double and then 2. So here the right option is D. See, each mitochondria is double membrane bound structure, you know already that one, dividing its lumen into two distinct compartments. One is perimitochondrial space and then one more is matrix. See here, this is the outer one, outer boundary, and then this one is the inner one, which is infoldings in the form of cisternae. So whatever this material is there, this distinct compartment is called as matrix, and this one is called as what? Perimitochondrial space. So here the thing is that each mitochondria is how many membranes? 2 membranes, correct, bound structure dividing its lumen into two distinct compartment. One is the matrix, another one is perimitochondrial space. 112. Matrix of mitochondria is option A, filled with a dense homogeneous substance. B, outer aqueous compartment. C, space present between the inner and the outer membrane of mitochondria. And D, present within the outer membrane of mitochondria. Just now I said about the matrix, which is filled with a dense homogeneous substance. 113. The outer membrane of mitochondria forms the dash limiting boundary of the organelle, while the inner membrane forms a number of dash. Option A, discontinuous and infoldings. B, infoldings and cristae. C, continuous and cristae. D, cristae and infoldings. Outer membrane is continuous, whereas inner membrane is having what? Cristae. So here the option C is correct. 114. Which of the following increase the surface area? A, matrix. B, inner membrane. C, outer membrane. D, cristae. Here the right option is D, cristae, because cristae will helpful for the expansion or else to expand surface area of the mitochondria whenever it is in need. 115. How many of the following statements are correct? 1. Only outer membrane has enzymes for ETS. 2. Only inner membrane has enzymes. 3. Outer membrane is devoid of enzymes. 4. Mitochondria matrix has enzymes of Krebs cycle. 5th, mitochondria is the site of aerobic respiration. And then 6. Matrix also possesses single-stranded DNA molecule and few RNA molecule. So here among them the correct answers are only 2. Outer membrane has enzyme for ETS is wrong, because it also has, inner membrane also has ETS enzymes. And here which one is correct means matrix also possesses single stranded DNA. No, matrix possesses double stranded DNA, which is circular. Mitochondria is the site of aerobic respiration is correct. Mitochondria matrix has enzyme for Krebs cycle is correct. Only these two are correct, remaining are wrong. 116. The matrix of mitochondria possesses single circular DNA molecule, a few RNA molecule, 70S type of ribosome, and components required for the synthesis of proteins. So among them only 2 of them is correct. or else possess, B, only 3 of them, C, all of them, and then none of them. So here the correct option is all of them. 117. Mitochondria divides by which kind of division? It divides by endomitosis, no. Meiosis, no. C, budding, no. D, fission, yes, correct. Now the questions from plastids. Plastids are the unique characteristics which are present in higher plants and some of the algae. 118. Plastids are found in A, only plants cells, B, only euglenoids, C, both plants and euglenoids, D, plants, euglenoids and cyanobacteria. So here the right option is both plants and some euglenoids, the plastids are present. This is the structure of plastids and the type of the plastids. Depending upon the function and then color, there are 3 types. Chloroplast, which is green in color, chromoplast, which will be having the pigmentations, leucoplast, which is the colorless, and then chloroplast is for photosynthesis, chromoplast is for pigmentation giving color, and then leucoplast is for storage. If you wanna keep this in mind, let me tell you a small trick. CCl, keep in mind CCl, chloroplast, chromoplast and leucoplast. Chloroplast is for photosynthesis, chromoplast is for pigmentation, leucoplast is for storage. 119. Classification of plastids are based on chromatophore, mesosomes, C, inclusions, D, pigments. Just now I said they are classified on the basis of what? Pigments. If it is green in color, it is called as what? Chloroplast. If it is colored, then it is called as chromoplast, and if it is colorless, it is called as what? Leucoplast. 120. Which of the following is responsible for trapping of light energy? Option A, chlorophyll like A, B, C, etc. B, carotenoids. C, chlorophylls and carotenoids. D, chromosomes. So here the right option is C, chlorophylls and carotenoids. Chlorophyll A is the main pigment which is helpful for the trapping of the light energy. Along with that, all the chlorophyll molecules and then carotenoids are responsible for what? Trapping of the light energy. 121. Carotenoids is group of A, chlorophyll pigments, B, chlorophyll and carotene, C, carotenes and xanthophylls, D, carotenes, xanthophylls and other pigments. So here the right option is D, carotenes, xanthophylls and other pigments. 122. Leucoplast is a unmodified plastid. B, contains stored nutrients. C, imparts colors to the plant cell. D, imparts colors to the cyanobacteria. So here the right option is contains what? Stored nutrients. CCl, just keep in mind CCl, chloroplast, chromoplast, leucoplast. Chloroplast is for photosynthesis, chromoplast is for pigmentation, and then leucoplast is for storage. 123. What are types of chloroplast? Number 1, chromoplast. Number 2, leucoplast. Number 3, amyloplast. Number 4, aleuroplast. Number 5, elaioplast. So here these are the types of plastids, but they are not the types of chloroplasts. So none of the above. 124. Elaioplast contains A, proteins and fats, B, fats and starch, C, fats and oils, D, fats, proteins and oils. Here the right option is option C, fats and oils. See, amyloplast, elaioplast and then aleuroplast. Next, 125. Aleuroplast contains option A, proteins and fats, option B, fats and oils, option C, proteins and starch? Option D, proteins only. Here the option is proteins only. Just keep in mind, wherever the word allero comes, then it means that protein. So here alleroplast means they have what? Proteins, whereas eleoplast have fats and then oils. Next, a major 126. Majority of chloroplast of a green plants are found in. Majority of the chloroplast of the green plants are found in. Option A, mesophyll cells of root. B, mesophyll cells of stem. C, mesophyll cells of leaf. Mesophyll cells of flower. So here the right option is mesophyll cells of leaf. Root won't have chloroplast, and then stem have few, because in young stem it looks green, isn't it? That is because of the presence of chloroplast. 127. Mesophyll cells are A, lens shaped oval spherical only. B, oval and spherical only. C, discoidal and ribbon shaped. D, none of them. Here the right option is B, oval and spherical shape. 128. What is the dimension of chloroplast? Dimension of chloroplast: A, length is 2 to 4 micrometer, and then width is 5 to 10 micrometer, and then B, length is 1 to 2 micrometer, and then width is 2 to 4 micrometer. C, length is 5 to 10 micrometer, and then width is 2 to 4, and then length is 2 to 4, and then width is 1 to 2 micrometer. Here the correct option is length is 5 to 10, and then width is 2 to 4 micrometer. 129. Number of chloroplast per cell may vary from dash per cell of chlamydomonas to dash per cell in mesophyll. So what is the number of cells may vary from in chlamydomonas to the mesophyll cell, he is asking. So here the right option is 1 per cell of chlamydomonas. Chlamydomonas has only 1 chloroplast, and then mesophills will be having 20 to 40 chloroplast. 130. Common features of mitochondria and chloroplasts are: A, number of membrane and type of DNA molecule only. Number of membrane and then types of DNA molecules only. B, number of membrane, ribosome type and DNA molecule only. C, type of thylakoid and genetic material, no. D, types of thylakoid, genetic material and permeability of the membrane. So here the right option is option B. The both chloroplast and then mitochondria has double membrane, and both of them have ribosomes. Ribosome type, both the ribosome type is same, and then DNA molecule is also, molecule of DNA molecule type is also same. Both have the circular small DNA. 131. What are types of thylakoid inside the chloroplast? A, intergranule thylakoid and stroma lamellae. B, granum thylakoid only. C, stroma thylakoid only. D, none of the above. So here thylakoid inside the chloroplast has two kinds of the thylakoids, that is integral thylakoid and then stroma lamellae. I will show you the diagram. So this is the chloroplast. When we take out only this part, and it will look like this. Let me show. So here this is granule thylakoid and this one is the stromal thylakoid. Stromal thylakoid is the connection between the adjacent granas. Okay, see, like this. This is grana and this one is one more grana. Whatever the connection between these two grana is there, no, this one is said what? Stromal thylakoid, because this thylakoid is present in the stroma. It is exposed to the stromal matrix. That's what it is called, what? Thylakoid stroma. And it is called granum thylakoid. Why? Because each one compartment is called as what? Thylakoid. Stack of thylakoid is called what? Granum, isn't it? So one unit when we are talking, then it is called what? The granum thylakoid. So This is called as integral and this one is called as stroma lamellae. Next question 132. Flat membranous tubules connecting the thylakoids in chloroplast is known as. Flat membranous tubules connecting the thylakoids in chloroplast is known as. Just now I said A. Granule thylakoid, B. Granum, C. Stroma thylakoid or lamellae, and then D. All of the above. So here thylakoid stroma or stroma lamellae is called what? It is the connecting the two flat membranous granum. 133. The membrane of chloroplast encloses a space known as. The membrane of chloroplast encloses a space known as A. Matrix, B. Cytoplasm, C. Lumen, and then D. All of them. So here it is called as what? Lumen. 134. The stroma of chloroplast contains 1. Enzyme for carbohydrate and protein synthesis, 2. Small single-stranded DNA molecule, 3. Ribosomes of 70S type. Here how many are correct, or else how many are having the chloroplast means only 2 of them. Enzymes for carbohydrate and protein synthesis and ribosomes of 70S type. Small single stranded DNA it won't have, it has a double stranded DNA molecule. 135. Chlorophyll pigments are present in A. Matrix, B. Stroma, C. Membrane, D. Thylakoid. Here the right option is thylakoid. The pigments are present in thylakoid membrane. 136. The ribosomes of chloroplast are A. Same as eukaryotic cell, B. 70S type with single subunit, no, C. 70S type with 2 subunits, D. All of the above. So here the right option is C. 70S type with 2 subunits. Next, ribosomes. 137th question. Which of the following statements are true regarding ribosomes? Number 1, granular structure. Number 2, first observed as dense particles by George Palada is correct. Third one, composed of mRNA and protein. Ribosome is only made up of proteins and some of mRNA. And then fourth one, surrounded by a single unit membrane. It is not at all made up of membrane. So fourth one is wrong. Only 3 are correct here. So right option is B, 3 of them. Next, 138. What are the types of ribosomes in a prokaryotic and eukaryotic cells? Types of ribosomes in a prokaryotic and eukaryotic with respectively. A. 70S, 80S, B. 80 and then 70S, C. 70 and 70S, D. 80S and then 80S. So here the right option, in prokaryotes they have 70S type of ribosome, whereas in eukaryotes they have 80S type of ribosomes. So next question, 139. How many subunits are present in a ribosome? A. 2, 1 large and 1 smaller subunit, correct. B. 3, 2 large and then 1 smaller subunit, no. C. Only 1 subunit and then D. 3. Don't go like this. See, don't waste time in these things. If you are very confident in your answer, just go with them, okay? Because they have, ribosomes have 2 subunit. You already know that one is large and then one is smaller subunit. So just go for it. 140. Subunits 50S and then 30S are found in 50 and then 30 are found in 60S type, 70S type, 80S type and then 90S type. So here the right option is B, 70S type. Okay, 60 plus 40 is present in 80S type. Don't get confused there. Okay, 50 plus 30 is having in 70s type. Most of the students will go with this option, but it is not correct actually. 141. What is the sedimentation coefficient? Option A, Swedberg unit, yes. B, measurement of density, correct. C, measurement of sites, yes, it is also correct. So option all of this is correct. Sedimentation coefficient means whatever we used to write 70s, no, 70s or else 80s for ribosome, that S means sedimentation coefficient. Okay, it is also called as Swedberg unit. It is used for the measurement of the density and then size of the ribosome. 142. What type of ribosomes are found in eukaryotic cell? Option A, 70s. Option B, 80s type only. C, both 70s and 80s type. And then D, 70s, 80s and then 60s type. Here the right option is option C, both 70s and then 80s type. Yes, of course. Why? Because in eukaryotic cells, the organelles like mitochondria and then chloroplast, they have 70s type, and then in in cytoplasm they will be having 80. Yes, one second. So question number 142. Question number 142. What type of ribosomes are found in eukaryotic cell? Option A, 70s type only. B, 80s type only. C, both 70s and 80s type. D, 70s, 80s and 60s. Here the right option is C, both 70s and 80s type, because 70s type of ribosome is present in organelles like mitochondria and then chloroplast, and then 80s type is present in cytoplasm, where it may be attached to the endoplasmic reticulum or else it may be in free position. So 142 correct option is C. Next, questions from cytoskeleton. 143. Cytoskeleton refers to the option A, cilia and flagella only. B, network of filamentous proteinaceous structure. C, microtubules only. D, both A and C. Here the right option is the network of filamentous proteinaceous structure. Cytoskeleton refers to the network filamentous proteinaceous structure of the cell. 144. Microtubules, microfilaments and intermediate filaments are constituents of option A, ribosomes. B, central sheath. C, cytoskeleton. D, cytolamellae. So here the right option is option C, cytoskeleton. Microtubules, microfilaments and intermediate filaments are the members of what? Cytoskeleton. 145. Cytoskeleton in a cell is involved in functions like, what is the function of cytoskeleton? Option A, mechanical support. Option B, motility. Option C, maintenance of the shape of the cell. And then option D, all of the above. Here the right option is all of the above, because cytoskeleton help in these all functions. Now the questions from cilia and then flagella. 146. Which of the following statement is untrue? It must be is, not in. Option A, cilia and flagella are hair-like outgrowth. B, cilia are small and works like oars. C, flagella are longer and responsible for cell movement. And then D, none of them. So here the right option is D, none of them. 147. Statement 1. Both eukaryotic and prokaryotic cells contain flagellae. Statement 2. Eukaryotic flagella are structurally different from prokaryotic flagella. So here both these statements are correct. Options we have like this. Both the statements are not correct. C, statement 1 is correct but the statement 2 is wrong, and then D, statement 1 is wrong but the statement 2 is correct. So with respect to that, option A is right because both the statements are correct. 148. The core of cilia and flagella is known as core of the cilia and flagella is known as A, central sheath, B, central microtubule, C, axoneme, and then D, bridge. Here the option C is correct. 149. The microtubule in cilia and flagella A runs parallel to each other, B forms the axoneme and outer membrane, C both A and B, and then D arranged central only. So here the right option is option A, runs parallel to each other. Next question, 150. What is arrangement of microtubules in the cilium and flagellum? In cilium and flagellum, what is the arrangement? A, 9 peripheral and 3 central, B, 2 peripheral and 9 central, C, 9 peripheral and 2 central, and then D, all peripheral. Here the correct option is 9 peripheral and 2 central. 9 plus 2 condition you will be fine. 151. The central sheath is option A, connected to inner doublet bridge, B, encloses peripheral doublet, C, connect to peripheral microtubules, and then D, all of the above. So here the right option is connect to peripheral microtubules. Next, 152. Which of the following statement regarding cilia and flagella are not correct? A, peripheral doublets are inner connected by linker. B, linker are also known as interdoublet bridge. C, both emerges out from a centriole-like structure, and then D, linker are also known as basal body. Here the right option is D, linker are also known as basal body. Next questions from centrosome and then centrioles. 153. Centrosome and centrioles can be found in A, animal cells only, B, plant cells only, C, both animal and plant cell, D, in plants and bacterial cell. So this is the centrosome which is only present in animal cell, which is a unique characteristic of animal cells also, and it will be helpful for the formation of what? Spindle fibers to hold the chromosomes during the cell division. 154. Centrioles in the centrosome are, these are the centrioles. Okay, these are the centrioles. Centrosome units are centrioles. A, parallelly arranged to each other, B, perpendicularly arranged to each other, C, arranged like a cartwheel, and then D, made up of triplets of centrally arranged microtubules. So here the right option is they are perpendicularly arranged in each other. They are perpendicular, you can see. They are perpendicular in arrangement. 155. The basal body of centriole has microtubular arrangement of the basal body of centriole 9 plus 0, 9 plus 2, 9 plus 3 and 3 plus 9. So here the right option is 9 plus 0. 156. The central part of proximal region of the centriole is, the central part of proximal region of centriole is A, known as radial spoke, B, known as central hub, C, connected to the peripheral doublets, and then D, all of the above. Here the right option is B, known as central hub. 157. The structure that give rise to the spindle fibers during the cell division in animal cell is A, cilia, B, flagella, C, both, D, centriole. Centriole is the right option. Now the questions are from nucleus part. 158. Nucleus as a organelle was first described by Robert Brown. Yes. 2. Stained by a basic dye. The material is known as chromatin by Robert Brown. No. 3. Stained by a basic dyes. The material is known as chromatin by Robert Brown. 3. Double membrane bound structure. How many of the above statements are not true about the nucleus and its material? So here the right option is only 1. This one, the stain is stained by a basic dye. It is nucleus is stained by acetocaramine, which is acidic, not a basic dye. 159. The nucleus has highly extended and elaborate nucleoprotein fibers known as elaborated nucleoprotein. First one, nucleoli. B, chromosome. C, chromatin. D, nuclear matrix. So here the right option is C, chromatin. 160. The content of an inner phase nucleus are nucleoli, chromatin, nuclear matrix, 2 membrane. So among them, which one is only having interphase nucleus is only 4 of the above. 1, 2, 3, 4 of the above are the interphase nucleus, content of the an interphase. Next, 161. What forms the barrier between the cytoplasmic content and then cell nuclear matrix? Nuclear matrix and then cytoplasmic content. What is the barrier? A, the outer membrane only. B, the inner membrane only. C, the perinuclear space, and then D, all of the above. So here the option D is right, because if I show you the zoom structure of the nucleus, this is the nucleus, this is the cytoplasmic content, and then this is the inner content, that is nucleus, nuclear matrix. The barrier between these two is inner mitochondrial layer, inner membrane of the nucleus also, and then outer membrane of nucleus also, and then space between these two, whatever this one is there. No, let me take a highlight or else pen. This one is the inner one, okay, and then this one bluish color is outer one, and whatever the space is there, that let me draw here. This is inner one, and then this one is outer one. This is inner one, this is outer one. This is inner one, this one is outer, this one is inner membrane, and then this one is outer membrane, and this one, whatever the pore I am showing, no, that is called what? Nuclear pore. Here what forms the barrier between the cytoplasmic content? This is the cytoplasmic content, isn't it? And this one is nuclear matrix. Nuclear matrix and then cytoplasmic content has a barrier which is outer matrix also, outer membrane also, inner membrane also, and then space between them, that is called as perinuclear space. So all of them is right. 162. The outer membrane of nucleus is continuous with the rest of the cellular organelles. 2. The inner membrane is continuous with ER. No, wrong. 3. There are interruptions known as pores present in outer membrane of the nucleus. Yes. 4th one, pores are formed by the fusion of both of the membrane. Correct. So here the right option is only 2. So only 1 and then 4th one is right. 163. The nuclear pores facilitates A. Movement of RNA and protein molecules in only one direction. B. Only proteins in both direction. C. Proteins in one direction and RNA in both direction. And then D. None of these. So here the right option is none of this, because the movement of molecules are both in both directions. 164. Few of the mature cells have no any nucleus? Option A, their function are not specific. B, are dead cells with cytoplasm. C, their function are controlled by some another cell. And then D, all of the above. So here the right option is their function are controlled by some other cells. 165 statement 1. The nucleus per cell varies per cell. 2. Normally there is only one nucleus per cell. So here both 1 and 2 are true, and then 2 is correct explanation of 1. Right option is B. Both 1 and 2 are true, but 2 is not the correct explanation of 1. 166. The nucleus matrix contains, the matrix of nucleus contains option A, nucleoplasm and chromatin. B, nucleoplasm, chromatin and mitochondria, wrong. C, nucleoplasm, chromatin and E are wrong. So here the right option is first one, nucleoplasm and chromatin. 167. What is not true about the nucleolus? A, spherical structure present in nucleoplasm, correct. B, membraneless structure. C, also known as ribosomal factory of cell, correct. So D, none of the above. What is not true? What is not true it is asking. So none of the above. All are correct about the nucleoplasm. 168. At which phase of cell cycle the nucleolus has a loose and indistinct network of nucleoprotein fibers known as chromatin? Option A, prophase. B, anaphase. C, interphase. D, metaphase. So here the right option is C, interphase. 169. Cells show structured chromosome during all phases except anaphase. B, all phases except metaphase. C, all phases except interphase. And then D, all phases except memphase. So here the right option is all phases except interphase. 170. Chromatin contains A, histones and non-histones and RNA. B, histones, non-histone proteins only. C, DNA and some basic proteins. D, both A and C. Here the right option is both A and C. 171. A human cell has approximately dash meters long thread of DNA distributed among its dash chromosomes. Yes, here the right option is B, 2 meters of long DNA we have in each cell of each nucleus of a cell of a body, and then they will be in 46 chromosomes. If I am talking in the pair, 23 pairs. 172. Each chromosome A, it has primary constriction. B, is visible only in dividing cell. C, has a disc shaped structure known as kinetochore. And then D, all of the above. So for the chromosome, all of the above are correct. 173. Function of centriole is option A, provides site of attachment to the spindle fiber on a chromosome. B, holds 2 chromatids of a chromosomes. C, both A and B. And then D, none. So here the right option is C, both A and B are correct for the function of centriole. Next, 174. What is type of chromosome having the middle centromere? See here I am giving the types of chromosome structure. If the chromosome is exactly at the middle, then it is called metacentric. If it is somewhat far, then it is called as submetacentric. If it is near to the terminal, then it is called as acrocentric. If it is totally at the terminal, then it is called telocentric. So here if it is having a middle center, then it is called what? Metacentric. Right option is metacentric. This is metacentric, this is submetacentric, this is acrocentric, and this is telocentric. 175. What is the type of chromosome having its centromere near the telomere? Near the telomere means this is the telomere. If the centromere is near the telomere, then it is called what? Telocentric. 176. Chromosomes having centromere slightly away from the middle is slightly away. This is the center. From slightly away from the center means what it is called? Submetacentric. Next, the chromosome having one long and one short arm are A. Metacentric and submetacentric, B. Submetacentric and acrocentric, C. Acrocentric and telocentric, D. Telocentric and metacentric. Here the right option is option C, acrocentric and then telocentric. 178. Function of telomeres in nucleus is 1. Poleward movement, 2. To initiate the RNA synthesis, 3. To seal the ends of chromosome, 4. To recognize the homologous chromosome. The telomeres function is to seal the ends of chromosomes. It seals the ends of chromosomes. 179. A non-staining is present on a few chromosomes, that is A. Secondary constriction or centromere, B. Satellite or centromere, C. Secondary constriction or satellite, and then D. None of the above. So here the correct option is C, secondary constriction or satellite, where there is a region where we won't find any kind of the staining material here. It will helpful for the study of the satellite DNAs. Next question is about microbodies, and this is the last question for today's session. 180. The membrane bound minute vesicle containing enzyme are known as membrane bound minute vesicle. A. Chloroplast, B. Mitochondria, C. Ribosome, and then D. Microbodies. The right option is microbodies. This is the end of MCQs, line by line MCQs of the chapter Cell, the unit of life, the one of the most favorite one. I can say this is my favorite also, because this chapter is the sole chapter, sole concept of the NCERT, I can say, because even though for the NEET and then even though for any of the competitive exam, you will definitely find at least one question from this concept. So this is the basic one, and this is the sole chapter, sole concept for your preparation. So don't forget the concept of this one, and then whatever the tricks, whatever the shortcuts I have gave for you. Next in next class I will be back with the next concept or else next chapter. So for today, bye bye.